Our body can be exposed to different types of pathogens such as viruses, bacteria, fungi, and parasites, which can cause illnesses and diseases. To defend against these pathogens, our immune system provides protection. White blood cells, B cells, and T cells are crucial components of the immune system that recognize and neutralize foreign invaders. Physical and chemical barriers such as the skin and stomach acid also prevent pathogens from entering. The immune system is a complex system that helps to protect the body from a wide range of pathogens. Adaptive immunity creates lasting protection against specific pathogens through antibody production by B cells and activation of T cells. It is characterized by specificity, memory, and diversity. Cell-mediated immunity involves T cells directly attacking infected cells and is crucial in fighting viruses and cancer. This type of immunity is mediated by helper, cytotoxic, and regulatory T cells.
Two major functional classes of T lymphocytes are CD8 positive cytotoxic T cell and CD4 positive helper T cell. But for this video, we will be discussing about the CD4 positive helper T cell. Helper T cell is known as CD4 positive T cell because it has CD4 positive glycoprotein on its cell surface. CD4 is a single-chain molecule consists of 4 immunoglobulin-like domains, which are D1, D2, D3, and D4. CD4 positive receptor recognizes antigen with MHC class II molecule and then activated. After being activated, CD4 positive T cell will scan for foreign antigens and stimulate other immune cells such as macrophages, B cells, and CD8 positive T cell to destroy pathogens detected.
Antigen presentation confined to MHC class II is required for CD4 T cell dependent immunological response. All APC interact particularly with different subsets of T cells that express the antigen-specific T cell receptor. MHC class II binds antigenic peptides produced by the proteolysis of self and non-self protein in endosome and lysosome. Present them to antigen-specific CD4 T lymphocyte. Recognition of peptide MHC class II by CD4 T cells promotes activation and differentiation into T helper cell subsets, as well as facilitating interaction between antigen-specific B cells and T helper cells. T cells are produced in the thymus and are designed to recognize a certain foreign particle known as antigen. They circulate throughout the body after leaving the thymus until they detect the antigen on the surface of antigen-presenting cells known as APC. The T cell receptor on CD4 helper T cells attach to the antigen, which is retained on the surface of the APC in a structure known as MHC complex. This causes the T cells to get activated for the first time. The CD4 molecules subsequently bind to the MHC molecule as well, so stabilizing the entire complex. The first binding between T cell specific for one antigen and the antigen MHC it match initiate the entire response.
After CD4 positive receptor binds to antigen presenting cell, CD4 positive helper T cell needs secondary signals to become activated. In this situation, CD28 molecule on CD4 positive helper T cell binds to one or two molecules on the antigen presenting cell, which are CD80 or CD86, and initiates proliferation of CD4 positive helper T cells, and to ensure the T cell is only activated by antigen presenting cell which has encountered a pathogen, while CD40 ligand on CD4 positive helper T cell binds to CD40 receptor on the antigen presenting cell, which help for the development of CD4 positive T cell dependent effector function. The bindings of CD28 to either CD80 or CD86 and CD40L to CD40 is called positive co-stimulatory interaction. However, to control the response, stimulation of CD28 by either CD80 or CD86 induces the production of CTLA4 on CD4 positive T cell, which it competes with CD28 for CD80 and thus reduces activation signal to CD4 positive T cell and lesser the immune response. Besides that, PD-L1 on antigen presenting cell binds to PD1 on CD4 positive T cell and induces the differentiation of CD4 positive T cell into FOXP3 positive regulatory T cell. The binding of CD8 to CTLA4 and PDL1 to PD1 is called negative co-stimulatory interaction. These two types of interactions on the integration within the T cell will identify the activation state of the T cell.
Cytokines are small proteins that are produced by various types of immune cells, including T cells, B cells, and macrophages, in response to an infection, injury, or other stimuli. Autocrine signaling occurs when a cell releases a signaling molecule that binds to receptors on its own cell surface, triggering a response within the same cell. This type of signaling is important in the regulation of cell growth, differentiation, and survival. Paracrine signaling occurs when a cell releases a signaling molecule that binds to receptors on nearby cells, triggering a response in those neighboring cells. This type of signaling is important in the coordination of cellular activities within a tissue or organ. CD4 T cells can differentiate into different subsets of effector cells depending on the cytokine environment and the nature of the antigen encountered. The major subsets of CD4 T cells include Th1, Th2, Treg, and etc. During an immune response, cytokine signaling can be amplified to enhance and coordinate the immune response through recruitment of more immune cells. Cytokines can attract other immune cells to the sites of infection or inflammation, which can then produce additional cytokines to amplify the immune response.